Hello and welcome. I'm your host, Neil Howard. Thank you so much for joining us for another segment of Health Professional Radio. We'll be speaking with Dr. David Beers in this segment, PhD, President and CEO at Halia Therapeutics. It's a clinical stage biopharma company developing novel therapies to improve the lives of patients with inflammatory disorders and neurological diseases. He's going to talk about the company's lead compound, which prevents the formation of NLRP3 Inflammasome complex and subsequently inhibits the production and release of pro-inflammatory cytokines. Welcome to Health Professional Radio, Dr. David Beers. Thank you for joining us this morning.
Thank you. It's great to be here.
Well, tell us a bit about yourself, a little bit of insight into your background, and what led to your vision of creating Halia Therapeutics.
Yeah, great. Yeah, so I have a PhD in cell biology. I learned very early in my career from my mentor, a guy named Dan Von Hoff, Dr. Dan Von Hoff, who was a clinical scientist, how to be involved in early drug discovery and early development. And that's really where the focus of my career has really been, is trying to discover new ways to develop therapeutics to benefit patients that are suffering from serious diseases. And I've spent the last 25 years focused on that. I've been fortunate to have discovered 16 drugs that I've taken into clinical trials, and I've worked on a couple drugs that have been approved, and I've kind of turned into a serial entrepreneur. I've started multiple companies, and usually those companies have been sold to other companies and worked for them for a little while, so I kind of gone in this cycle. And Halia, we started almost 5 years ago. And the vision for Halia was a little different than other companies that I started. It really started with one of our co-founders and I getting together, looking at some of his data that he had been generating in the area of Alzheimer's disease. So his name is Keone Kawe, and Keone is a human geneticist, and he had discovered when he was a postdoctoral fellow, he was part of a large consortium of researchers at Washington University in St. Louis, and he had discovered as part of this group a number of genetic risk factors that put people at risk for developing early onset Alzheimer's, other forms of dementia. And when he moved from Washington University back to Utah, where he's from and where I'm from, that's where we are now, is in Salt Lake City. He had identified a family that had a high-risk genetic polymorphism. So he had a DNA sequence that put them in a gene that put them at high risk for developing early onset Alzheimer's. And when he was studying this family, he discovered that to his surprise, that nobody in this family actually had Alzheimer's, even though they had this bona fide, well-identified genetic risk factor. And so he thought, well, there must be something else protecting them from Alzheimer's, that some other genetic mutation or polymorphism that protected them. And so we started Halia with the idea of trying to find out what that was and seeing if we could develop a drug that would mimic the protective effects of that particular mutation. And so that's what we ended up doing. So we were able to identify that this family had a second polymorphism or mutation that actually provided them protection from the other risk factor that they had inherited. And when we discovered the gene that was that they carried, the second protective mutation was in a gene called RAB10, and RAB10 happens to be part of a protein trafficking system inside the cell. So it's involved in moving proteins to the right places inside the cell. And so that was very interesting to us, and we started to dig into the biology, and we eventually discovered, just probably like everything that I've been involved with, you start out with one idea of where you're going to end up, and you end up someplace different, and a lot of times it's better than what you originally thought. No. And we ended up finding a new way to target chronic inflammation, and that's really what the focus has been on with Halia.
Well, what are some of the current approaches that exist to target this chronic inflammation?
Yeah, it's been an interesting challenge for years. When I was a young graduate student, I took an immunology course, and they taught us that chronic inflammation was just the failed resolution of acute inflammation. So acute inflammation is a normal, healthy response to damage or injury that happens to our bodies. And, our thinking back then was chronic inflammation was just an acute inflammatory response, got stuck in the on position and never turned off. And what we know now is that there's a whole process that the cell can activate chronic inflammatory signaling that's separate from it shares some of the same components, but it can be distinctly different than acute inflammation. So for years we've tried to treat chronic inflammatory diseases by inhibiting the signaling downstream of the source, the site of injury. So things like COX-2 inhibitors or anti-cytokines like Humira is a very popular drug. It's done very well, and it takes out a particular signaling molecule. But what we've discovered, and we meaning the whole field, not just us, but we certainly have contributed to it, is there's actually a machinery inside the cells that are producing the signal, and that machinery is called the inflammasome, and specifically the NLRP3 inflammasome. And we've figured out a way to turn that off. And when you turn that off, the whole signaling pathway gets shut off. So instead of just taking one piece of it out with a therapeutic, this is the first time that we think we can get to the root cause, which is this big protein complex, this machine inside the cell, and completely turn that thing off.
HT6184, I believe that is the lead program, this lead compound that targets inflammasome. Talk about this. Describe how this compound uniquely solves that problem.
Yeah. So when we had some insight from this discovery that we made about the protein trafficking component of the inflammatory response. So in order to coordinate this assembly of this big giant protein complex inside the cell has to move a lot of proteins to the right place at the right time. And what we discovered is there was a particular location that the cell was assembling this big protein complex. And we thought, well, there must be something important in that location because all the other proteins are being brought to this one place. And so we figured out what that was, and it turns out there's one of the components of the inflammasome, a protein called NEK7, NEK7. And NEK7 is kind of the glue that holds the whole thing together. It allows the components of the inflammasome to interact with each other. And we thought, well, if we could inhibit the activity of NEK7, its ability to kind of glue these things together, maybe we could stop the assembly and maybe even promote the disassembly of the inflammasome. And so we went to work designing a drug that could prevent the function of NEK7. And it works through a mechanism that we call an allosteric effect, which means that it changes the shape of the protein, NEK7 protein, and when you change the shape, you change the function. And so that's how our drug works. It's the first drug to be designed to have this particular function. And we've tested it in over 20 different models of inflammation to show that it can block the assembly of the inflammasome complex and can actually promote the disassembly of the complex. And so we can kind of turn that switch off, the chronic inflammatory switch. And that's really what people have been searching for many years is how do you turn that switch off once it gets turned on? And we think we may have a way to do that.
Well, are there any upcoming milestones for Halia? And give us a website where our listeners can learn more.
Yeah, that's great. So we have we're just completing our initial clinical evaluation of the compound. And so typically what we do is we run a safety study first. It's phase 1 clinical trial. And so we're just wrapping that up. So the first part of that, we did what's called a single ascending dose where we give just a single dose of the drug. We watch how the drug gets absorbed and eliminated. And then we do multiple ascending doses where we give individuals multiple doses over a 2-week period. And we watch how the drug gets absorbed and eliminated. We look for any potential safety signals. And in our case, we were able to build into the study some measurements of immune cells and how they're being affected by the drug as individuals take that. And so we've got a lot of information. We're wrapping that study up. We should be done just here in a couple of weeks. We've treated about 90 individuals so far, and so we've got a good amount of data to help us design and to look at the next clinical trials, well, we'll really try to look at specific chronic inflammatory diseases. And we're interested in many different diseases, including inflammatory bowel diseases like ulcerative colitis, Crohn's disease, inflammation in the liver, in the lungs, in the cardiovascular system, and really where we started back in the brain in neuroinflammatory diseases like Alzheimer's and Parkinson's. So we're excited about the direction that we're headed, and if people want to keep an eye on what we're doing, our website is www.haliatherapeutics.com.
It's been a pleasure speaking with you today, David. I'm hoping that you'll come back and tell us how things are going as clinical trials continue.
I'd very welcome that, so I look forward to it.
You've been listening to Health Professional Radio. I'm your host, Neil Howard, in conversation with Dr. David Beers. Audio copies of this program are available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to our YouTube channel at youtube.com/Health Professional Radio.